First of all, welcome everybody to the GHGA webinar. Today, we're going to be hearing an introduction to computational immunology. I'm so sorry, the title is wrong here. We're talking about computational immunology with Katharina Imkeller today. Before we get started, just a couple of quick notes. One, we are recording this meeting, so you can watch it back in a couple of weeks' time on our YouTube channel. However, none of your video or audio will be included. We will be stopping the recording before we get to the Q&A session at the end, so you don't need to worry about that. And before we get started, I want to briefly introduce GHGA to you. It is short for the German Human Genome-Phenome Archive, and our mission is to provide a platform for GDPR-compliant human omics data sharing. And that is in order to enable the secondary use of clinical omics data in research for biological discovery and replication of findings. And this will help translate research insights. And since this involves interactions between very diverse communities, like researchers and clinical researchers, patients, scientists, and physicians, we would like to see this webinar as kind of a platform to exchange ideas from different groups. And if you're interested in any of our other training resources, I invite you to visit our website. We have all of the training made available to you on demand as well in different categories, as well as on our YouTube channel. You can see all of the recordings from previous webinars. And now I want to briefly introduce our speaker today. Katharina has a Master of Science in Molecular Medicine from the Charité University in Berlin, and has been a PhD student with the Max Planck Institute for Infection Biology in Berlin and the DKFZ in Heidelberg, and has been a postdoc with the DKFZ in the EMBL at Heidelberg. And since 2021, she is a junior group leader in computational immunology, affiliated with the MSNZ, FCI, and Edinger Institute at the University Hospital in Frankfurt. And with that, I would hand over to Katharina and let her talk to you.
I'm very happy to be here, and I would like to give you an introduction about general concepts and methods in computational immunology. And I would like to start with some general characteristics of immune responses that one has to keep in mind when analyzing data from immunological context. So the first important characteristic of an immune response is that it has to be coordinated in time. So it has to be fast after infection, but it also has to be temporary or acute, and the immune response has to stop at some point. So this is the first challenge the immune system has to master. It has to coordinate itself in time. Second, the immune system has to coordinate itself in space. So you want the immune response to take place in the tissue that is infected, for example, but at the same time, cells from infected tissue need to migrate to other organs, such as lymph nodes, and other cells from the lymph node may need to migrate towards the tissue that is infected. And this coordination is very important so that the immune function is maintained. The third element that I would like to highlight is antigen specificity and memory, which is a characteristic of the adaptive immune system. And it is important for in vaccination, for example, where the adaptive arm of the immune system, so B cells and T cells, can recognize particular antigen using their surface receptors. They have a specific antigen. Each cell has a specific antigen they can recognize, which makes the immune response antigen specific. And if you re-encounter the same antigen again, there's a memory response that can be reactivated. So all of these three elements are important. And I would like to show today a few computational methods on how to study these kind of coordinations and interactions. The data that I'm covering today is from different molecular and cellular characterization methods for immune responses. So there's spatial transcriptomic data, which is the quantification of gene expression in a tissue with spatial resolution. Then there's single-cell transcriptomic data, and there's also B and T cell receptor repertoire data, so basically the sequences of the B and T cell receptors. And I would like to show how to integrate these three different data layers in order to model the immune interactions in the tissue and in order to understand the immune function ideally. So what you will learn today are computational approaches from different areas of the immune function. So they range from immunogenetics, how the genes in our genome encode the receptors and ligands of the immune system, how these receptors and ligands then mediate cellular interactions, and how these cellular interactions shape the immune function in the tissue. And in the webinar today, so we are going to have three different parts, and we are going to start with the broad concept of immune function, and we will look at cell types and phenotypes. After that, we will look at how to characterize the interactions of immune cells, how they use the receptors and ligands to interact. And finally, in the last part of the talk, which will also go into a bit more detail than the other two parts, we will look at how antigen specificity is defined and encoded in the immune system. So let's start with the cells and phenotypes. And what immunologists typically do in order to define cell types are flow cytometry-based experiments. So these experiments, you take cells from a tissue or from the blood, and you label the cells with fluorescence antibodies that can recognize surface markers on the cells. And then you analyze the data. You can set gates by segregating different expression patterns from each other, and you can thereby identify the cell types of interest, such as NK cells, B cells, T cells, all kinds of different immune cell types that exist. So this is how it is typically done and how it was also historically done. Nowadays, in immune research projects, typically the data does not only or not at all stem from flow cytometry experiments, but rather from single-cell transcriptomic experiments. And I would like to discuss how the cell type assignment can be done in single-cell transcriptomics analysis workflows. I would like to say right away that, so I'm not going to go into too much detail about the analysis workflow, and I would like to warmly recommend the previous seminar by Florian Heil, who discussed single-cell RNA analysis workflows. And I would like to just to highlight two steps. So in the whole analysis workflow of single-cell RNA analysis, the step that we are talking about now, the cell type assignment, is done after the pre-processing normalization at the time when you perform clustering and annotation of the clusters. And one thing to keep in mind is that the cell types that will be identified using single-cell RNA sequencing data will probably not 100% be the same as the ones that would have been identified using FACS markers or flow cytometry, because the expression of the surface markers on the cells is not 100% related to the expression of the genes in the cell. So nevertheless, let's see how this looks like. So here I have analyzed for you an example data set from immune cells from the blood. And I performed clustering. You see that there are different clusters. And these clusters, when you look at the expression of particular marker genes that are known in immunology, you can see that, for example, the green, red, and purple clusters here are probably T cells because they express CD3. These ones here are probably B cells, and you see that there's already a certain pattern and it's not so difficult to interpret. However, if you have very large data sets with multiple donors and maybe also from more complex tissue, and you also want to go into detail about the annotation, people try to use automated cell type annotation. And these automated cell type annotation approaches typically use cell type reference datasets, so datasets that are already annotated. And then they use these references to annotate your single cell, so the new single cell sequencing data. And there are two general approaches which differ in the selection of features that are used for the annotation. So you can either use data-derived marker genes, either derived from your reference data or from your own single cell sequencing data, or you can also use the full gene expression profiles. If you do this, it's important that the full gene expression profiles mean the same thing in the reference and in the actual data, obviously. I listed a few tools that can be used for automated cell cluster annotation, and I would also like to recommend further reading if you're interested in this topic from these two reviews here. So what I did on the next slide is to use this tool, single R, to annotate the data set that we saw on this slide. So let's go to the next slide. You see that now, so I used single R and 3 different reference data sets. So this is one reference data set, this is one reference data set, and this is one reference data set. And you see that although I use the same data that I want to annotate, the annotations that I get are slightly different. So you see that T cells are more or less still recognized correctly, but there are differences in terms of how granularly the T cells are annotated, whether you have differences between CD4 and CD8 T cells. And you also see that in some of the annotation examples, there's also confusion between some types of T cells and NK cells. So this is something I think it's important to keep in mind, that the reference that you're using for the automated cell type annotation counts. And it is always good to use a reference that is close to the tissue and to the question that you're interested in, because the cell types also differ between tissues. So with this, I would like to move one step further and also mention that you can integrate single cell data with spatial transcriptomic data. And these computational methods basically have the aim to map the single cells to the spatial location. So to bring the single cells from your single cell sequencing data into some kind of spatial context. I selected one article here which introduces the cell to location tool. And the cell to location tool helps you to, so starting from single cell and spatial transcriptomic data, it helps you to find the most likely location of the single cells within the spatial organization of the tissue. There are also other popular methods that can be used. And you can also use, so if you only measured one of the data, if you only have spatial transcriptomic data, it's also possible to use public reference data sets for the mapping. This mapping is also sometimes called deconvolution, where you basically in each spot of or in each area of the spatial transcriptomic, you deconvolute which cell types were present there. With this, I would like to formulate the take-home message here for this part of the talk, which is that so cell type annotation is one of the first steps and also one of the most important steps in a typical single-cell immunology project. And it is also prerequisite typically if you want to perform downstream analysis. The spatial context of the single cells can be interrogated by integration with spatial transcriptomic data, which is also commonly done. About the automated cell type assignment, I would like to mention that it works well for common cell populations and also whole transcriptome sequencing. It does not work so well if you enrich your study for rare cell populations which do not have good reference data sets. And again, recommendation, if you're interested in more details about the single cell part here, take a look at the other webinar. So now moving on to the second part. And now that we have understood how to annotate the cell types, one of the next steps could be to look at the interactions of the immune cells. And so there's a whole large area of research that is conducted about ligand receptor interactions and tools how to annotate and quantify ligand-receptor interactions in single-cell data. And all of these resources are based on databases of ligands and receptors. So these databases contain information on which ligands interact with which receptors. I list a few here. If you're interested in the topic, there's also a nice review that I can recommend. And please note that here I'm only talking about the receptors and ligands that are encoded in the germline. We will later in the third part of the talk also have receptors that are not encoded in the germline. So these databases listed here can be utilized in workflows where you score the receptor-ligand interactions. A typical workflow for scoring receptor-ligand interactions starts from an expression matrix. In our case, an expression matrix could look like a single-cell expression matrix where each column belongs to one single cell, and the entry in the matrix determines or indicates how at which level a particular gene is expressed in a particular cell. So you can combine this expression matrix with the database of receptors and ligands that interact. The next step is to filter your expression matrix only on the ligands of receptors of interest that are part of your database, and then use this filtered expression matrix in a statistical framework where you perform the scoring of the ligand-receptor interactions. So the scoring is done using a scoring function. We will take a look at scoring functions in the next slide. But what all scoring functions do is to take the information from the expression and the information from the receptor-ligand pairs and output a score of each receptor-ligand pair. And this table of scores is then used for further interpretation and visualization. So now let's take a more detailed look at the scoring function. The scoring function can be very simple. The first one here, so here I showed two examples of scoring functions. The first one is simply an expression thresholding, where you look at the expression of the ligand, you look at the expression of the receptor, and if they are expressed above a certain threshold, they are labeled as this molecule is expressed in the cell. This is the table that you get out. So you would have binary entries for presence of ligands and receptors, and you can then calculate the communication scores, which is also binary, and it's only positive or only one if both ligand and receptor are expressed. So this is a simplistic approach. A more complicated approach is expression products, where you multiply the expression values of the ligands and receptors, and you then get a continuous distribution, continuous values for the communication scores. For more complete list of tools and methods, please refer to the reference shown here. Now I would like to show you one example of how this receptor-ligand scoring can be integrated into research projects. So this is a publication from 2018 showing the interactions of receptor-ligand pairs on different cell populations. So you have clusters of cell populations here in blue, you have other cell populations here in red, and you, in the dot plot here, you have the interaction score of the molecules in blue expressed in the blue and in red expressed in the red cell types. And you see that some cell types are likely to interact via certain receptor-ligand pairs based on the scoring that was previously done. So you can use this method to compare interactions between cell types. You can also use it to compare interactions between conditions. So if you have a treatment condition, for example, and a non-treatment condition, you can see whether the treatment changes the interaction scores. So lots of things that can be done with this approach. So now I would move on to the third part of the talk, which will be a bit more detailed. Oh no, sorry, I still have take-home messages for you for the second part. So the what I would like to say about receptor-ligand interactions is that the choice of the database matters, and you have to be aware that there are specific receptor-ligand interactions databases that have been developed by specific research communities, and depending on what your question is, you have to look for the right database. So it also matters whether you're using mouse or human data. So you have to carefully select here. You can also create your own database, or you can modify existing databases and use your customized database in the existing statistical tools. Common problems for the workflows are the redundancy of interaction. So one ligand can interact with many different receptors, and one receptor can interact with many different ligands. So this comes down to the quality of the database, basically. And if you want to identify really functional differences in their interactions, typically you need to do an extensive follow-up. So now that we have seen the take-home messages for the second part, let's move on to the third part for the antigen specificity. And I would like to start this part with a picture of the molecules that mediate adaptive immune responses. So these are the adaptive immune receptors, and the interactions shown here are specific for particular antigens. So the first one you see is an interaction of an MHC class 1 molecule with an antigen and a T-cell receptor. So the MHC molecule presents the peptide antigen here in purple to the T-cell receptor, and the T-cell can get activated. The same is the case for MHC class 2 molecules and peptide antigens. Here the structure of the molecule is a bit more complicated, but it can also present peptide antigens to T-cell receptors. And finally, here on the right, the third one is a depiction of an antibody-antigen interaction. You have a part of an antibody that is binding its antigen, and you also see that the antigen here is not a peptide antigen, but a more globular antigen. And so this part about antigen specificity, now I would like to start with the MHC part. As I mentioned, so here in green and orange, the MHC molecules are shown, which can present peptide antigens to T cells that recognize their cognate antigen via the T cell receptor. And you can imagine that these interactions have to be diverse. So there has to be, from an evolutionary perspective, the possibility to recognize a large repertoire of different peptides. And this is so on an individual and also on a population level, this is ensured by allelic diversity of the MHC molecules. What you see here on the left is the surface or the peptide binding cleft of an MHC molecule which binds a peptide. Here the peptide is shown in yellow. And the allelic diversity of the MHC molecules is often affecting the residues that form the peptide binding cleft. So, and each exchange in amino acid of the MHC molecule in the peptide binding cleft leads to a different repertoire of peptides that can be bound and presented. To give you some numbers, currently there are, for MHC class 1 molecules in humans, there are several thousands for each of the A, B, and C genes, several thousands of different alleles that are known. So from a bioinformatic and computational point of view, it is interesting to leverage resources that help you annotate those alleles. And I would like to introduce a few resources that can be used for this. So the aim of these resources is always to help you deal with the huge diversity of alleles, and also give you references of the alleles that are known. There are several bioconductor packages that can be used. They have different functionality, and they typically utilize databases for HLA or MHC alleles and peptides. I have 3 different databases listed here. So the first one, for example, contains the sequences of the alleles that are known in humans. The allele frequency net database contains the distribution of those alleles and their frequency in human worldwide populations. And the third one is a database that also contains data on interactions of MHC molecules and peptides. One of the reasons why you would maybe like to use these resources is when you have a computational workflow that aims to predict peptides that are presented on the MHC molecules of a particular donor or of a cohort of donors. So this application here uses computational prediction tools that allow you to predict which peptides are presented on which MHC molecules. And a typical workflow would look like this. So you use your genomic data from your patient cohort to annotate the HLA alleles and to type the HLA alleles. For this, you can use the resources that I've mentioned before, and you get the names and the protein sequences of the MHC complexes that are present in each of the donors. With this, you can then use MHC binding prediction tools. So these are computational tools that, as I said, predict which peptides are presented on the MHC molecules. This is important for cancer immunology. For example, if you want to detect neoantigens, it's important for vaccination, also for a virus infection. Just to mention how these MHC binding prediction tool work, they typically take as an input the peptide and the MHC sequence, and then they have some kind of computational box that scores those interactions and predicts a binding score of each peptide with each MHC molecule. Further recommended reading is this review here, which introduces several different tools for the prediction. Now let's go back. So we've seen the antigen presentation side of things. Let's go back to this overview of the adaptive immune receptors, and I would like to focus now on the T cell receptor and the B cell receptor or antibody side. So these T cell receptors and antibodies are the molecules that can specifically recognize a particular antigen that is either presented on MHC molecules or that is present as a globular antigen. And so I just plugged in the power of my computer. It's important to notice that the T cell receptors and B cell receptors that I depicted here, that are not encoded per se in our germline genes. But there's a complicated process that is taking place during the development of the cells. It's called VDJ recombination, and the most important role of this process is to allow encoding of functional T cell receptor or B cell receptor chains, and also to introduce sequence diversity into those antigen receptors. Here you see an overview of the process of VDJ recombination. So in our germline, there are multiple different V, D, and J. So these are the names of the genes. There are multiple of those present, and they can. There's a process that is involving rearrangement of DNA that then leads to the joining of one V, one D, and one J segment. And this then encodes a functional protein chain. The mRNA molecule would look like this. So in blue, you have the constant region of the antibody or the T cell receptor, and in orange, you have the variable region, so the one that carries the sequence diversity. The mRNA molecule encodes both the variable and the constant regions, and it also, the mRNA molecule gets translated to an antibody antibody molecule in this case, which is shown here. It has multiple chains, so there's a heavy chain and a light chain. I only show the recombination process of the heavy chain, but the recombination of the light chain is similar. And what is shown here in yellow or in brighter orange are the CDRs, the complementarity determining regions. And these are the regions that are typically binding to the antigen. So I've shown you how this VDJ recombination process works. And I've mentioned that there are multiple different V, D, and J genes that can be recombined. Here are some numbers for the human heavy chain. So 45, 23, and 6 different known genes according to the IMGT database that is the reference database for these kind of sequences. And if you imagine that one V and one D and one J can be randomly selected, you already see that there's huge level of combinatorial diversity. There are some other processes. I don't want to go into too much detail. Maybe just to mention that the recombination here involves addition or removal of nucleotides, which further increases the diversity of the sequence. So now let's look at this slide here. This is a representation of how diverse the antibody-antigen interactions can be. So these are schematic representation of how variable regions of antibodies can interact with the antigen. In gray, you see the part of the antibody. In blue, you see the antigen. The second column is the structure representation taken from crystal structures. And in pink or yellow, I highlighted the residues that interact with the antigen. So you see the structure of the antibody molecule, and in pink, the residues of the antibody molecule that interact with the antigen. And here in the last two columns, so the first one is for immunoglobulin heavy, so the heavy chain of the antibody, and the right one for the light chain of the antibody. And again, I transferred the pink labels to where approximately they locate in the sequence. And you see that, so what I mentioned before, there are some regions which are more often interacting with the antigen. Here, for example, this is the complementarity determining region 3, but there are also some other regions. And I hope that this image gives you an idea of why it is so important to annotate the immunoglobulin sequences and to understand where the V, D, and J segments that recombined are located here in this complete sequence. So it's related to germline polymorphisms, for example. So what is the germline gene that is utilized? Are there polymorphisms that would be beneficial to raise a specific immune response? So it's basically related to the donor genetics. And now that we have seen the structures, I would like to illustrate of how an annotation workflow can be done. So how do you annotate the immunoglobulin sequences? This is just a small portion of an immunoglobulin sequence that I annotated with a tool called IgBlast. And what the tool does is to align your sequence of interest to the reference database of V, D, and J segments. And then it also annotates the complementarity determining regions. These are the features that I mentioned here. So these are features that are important for annotating immunoglobulin and also to some extent T-cell receptor sequences. There's an additional feature that I would like to mention, which is the somatic hypermutation feature. So the molecules, the DNA encoding the immunoglobulin molecules can mutate within a B cell in order to increase the affinity of the B cell receptor to its antigen. This is shown here. And the way we annotate those somatic hypermutations is by comparing your sequence of interest to the reference database. And obviously, if there's a difference, you know that there was a mutation. And this mutation is typically located in the antigen binding site. I list two tools and infrastructures for annotation of AIR sequences. So AIR here stands for Adaptive Immune Receptor Repertoire, which comprises both B and T cell receptor repertoires. And I would recommend them if you're interested for further reading. So now I have a few other things to mention about those adaptive immune receptor repertoires. There's an AIR data standard format defined by the community. You can find it here. And its aim is to make things interoperable. So if you want to transfer data from one tool to the other, this structure allows it. And also to standardize the information that is given for immunoglobulin or T cell receptor sequences. A lot of people are using CellRanger for their annotation of sequences, and the file that is in AIR standard format is the AIR rearrangement file. In the last 2 slides, I would like to go back to this idea of integrating different sources of information. And I would like to highlight one particular example, which I like very much, about how to integrate single-cell transcriptomic and B and T cell receptor sequencing. And this is based on methodology, which is offered from different companies, or which you can also design yourself, where you sequence at the same time single-cell transcriptomes and the belonging B and T cell receptor repertoires. And of course, you do this for B or T cells because the other cells don't have a B or T cell receptor. And this allows you then to include the information about antigen specificity within your model of the immune reaction of interest. And I would just like to show this, this example here from this publication where the T cell receptor sequences and the transcriptomes of the T cells were measured at the same time. Here's some annotation of cell types. So CD8 T cells would be here, CD4 T cells somewhere here, and regulatory T cells here. And by using the information from the T cell receptor, and by knowing that one T cell receptor is so specific that it is very likely that each cell that expresses this particular T cell receptor is related within a cluster. So this is what is called clonotype. This is like clones of cells originating from the same original T cell that had this T cell receptor. And yet you can map those clonotypes onto the expression space of your single cell data, and thereby see whether cells can change their phenotype or whether particular antigen specific T cells have a particular phenotype. So this is a very resourceful method to use. As a summary, so we've seen how to annotate immune cells, how to look at the interactions using receptor ligand databases. For the antigen specificity, I've talked about the annotation of HLA or MHC diversity and the annotation of adaptive immune receptor repertoires. If you're interested in immunological data sharing, so if you're generating immunological data or would like to use immunological data, or if you develop tools for immunological data, I recommend to take a look at the NFDE for Immuno community and consortium, which has the aim to harmonize data representations for immunology, promote the adoption of FAIR principle within the community, build and operate repositories for data, and also develop tools that facilitate standardized and reproducible analysis. Here's the website. You can also follow the consortium. And with this, I would like to end the presentation. Here's some further readings as references. And I'm happy to take questions.